Hello there, this is Dr. Dan Guerra coming to you from Authentic Biochemistry Studios in the inland Pacific Northwest of the beautiful USA. Today is the 16th day of August 2020, and we are moving closer and closer to when school starts. So looking forward to that for sure. And to prepare you for that, I like to do what I always do, which is provide lectures, because school lectures kind of go together. You know what I mean. So let's get right into this discussion, shall we? All right. So as I said, it's Authentic Biochemistry, a video podcast for today. That's me, Dr. Dan Guerra, and I'm glad to present this to you. This is work that I've been doing for some time now. It's in association with my biomedical consulting. These lectures are free, as is my podcast, which is called Authentic Biochemistry, oddly enough. And you can reach that just by going and typing that in for finding podcasts, because I've tried it and it works. But also you can go to my YouTube channel, and that's where you're going to be viewing this. Or go to my Facebook page, and if you go to my Facebook page, you're going to find all the audio and all the video, and it's all for free. However, it would be exceptionally good if you would consider joining my Patreon page and giving some kind of contribution to these lectures. It would be helpful to me tremendously, and also once I set this thing up with Patreon, I can do some extracurricular activities that make it more of a interesting journey for those who become subscribers and patrons. Just like the people in the Renaissance, and even during the 19th century in Europe, scientists as well as artists had patrons. And sometimes universities pay your salary. I'm retired from that now. And sometimes patrons pay your salary. And that is nothing wrong with that, because it doesn't alter at all the content. I don't bias anything. What we do here in Authentic Biochemistry is we go into the scientific literature, published peer-reviewed literature, of which I've contributed and still contribute to, and we discuss what's published in those papers relative to a very specific topic. And we discussed at the level of looking at data and determining whether or not the data actually can be understood as evidence, explain the evidence within the backdrop of the architectonic of what research is being done in that given area, and then see whether or not it coheres or is foundational or rebuts what has been published, and then look to see whether or not there are ways to see this as a contribution which may alter the viewpoint and the overall stasis of the scientific endeavor under consideration. So what I've been talking about the last 13 episodes. That's including the Authentic Biochemistry podcast, which are only about 30 minutes in length, and then the videos, which are about an hour. It's about, I think we're at number 13 now, is I've been discussing the general arbitration of senescence or aging in humans. And we talked a lot about human aging at the very beginning of this arc, and then we talked a fair amount about the molecular genetics. And then we got into some cell physiology, some cell pathophysiology. And then about the third or fourth episode, I started doing compare and contrast between senescing or aging and then the process of oncogenesis or tumorigenesis. Tumorigenesis being runaway growth, of course, eventually leading potentially to high morbidity or even mortality in human disease. Comparing and contrasting that to the aging process, where the cell starts to shut down its anabolic machinery and goes into a senescent phase where it can still be reprogrammed to be immune active, even to the point of hyperimmune active. So I know you hear often, because of biomedicine being discussed a lot in the news, is that people who are immune compromised are ones that are susceptible to illnesses. While immune compromised, meaning, for example, a deficit in the ability to launch an immune response, either at the innate or acquired level, can lead to more infections. I think quite the contrary. If you have a hyperimmune response, that's what gets you really ill. And in fact, the autoimmune diseases all have to do with inappropriate or hyperimmunity. And then responses even to infectious agents often make you more sick, put you in a high morbidity, high fever, even coma and death because of the response to the infectious agent. And that's all a hyperimmune response. So I want to clear that up in case you haven't picked it up in all the times I've been discussing it. So where we're at now is we're discussing mevalonic acid pathway, the MVA pathway. Of course, that's on the road to de novo cholesterol synthesis or cholesterologenesis. So you know I'm a lipid biochemist, and you know that cholesterol is a prenylipid. And so I want you to understand that cholesterol isn't just a component of the membrane. It isn't just a precursor to bile acids and to steroid hormones. The intermediates in the biosynthesis of cholesterol play a major role in signaling and control of metabolism down to the level of bioenergetics and cell division, or cell quiescence, or cell autophagy, cell programmed death, such as in apoptosis, or even that rapid cellular proliferation that we see either in, for example, the T cell or B cell response, or unfortunately also in cancers. Okay, so let's get back to this. Again, 16th of August. So I got this from a really good site that I recommend to everyone. It's called Immunopedia. So it's a place where you can get good deliberative descriptions of immunology at the medical and even at the scientific level. So the medical level is one level, but the scientific level is a much higher bar, because medicine is not so much a science as it is a procedure. Whereas science, what we're trying to do there is be absolutely aware of looking for exojective descriptions of nature. And we do that by finding evidence, demonstrating whether or not that evidence can lead us to anything that's close to truth that we can believe, and then knowing that those are only benchmarks on a step gradient, a step pyramid leading higher and higher and closer to what we think nature really does provide and what we hope to be able to comprehend via our executive decisions. So anyways, a good plug for Immunopedia. So let's see what it says in general. I'm giving you a general description of a couple of things. First of all, just the acquired immune response. So lymphocytes engage teleologically, that is, they display specificity. So they have a telos, they have a goal. They also can be quite heterogeneous, and there is memory cell production in the lymphocyte brigade of the leukocyte common immune response. There are two major classes involved in recognizing the antigen, usually some kind of foreign protein, but it can be a foreign carbohydrate or a foreign lipid, or any combination of the three major macromolecules. Of course, it can also be a nucleic acid. Number one is the thymus-dependent or T lymphocyte, which participates in cellular responses against intracellular pathogens, organ transplants, and malignant cells. Okay. Then there is the bone marrow or bursal-dependent B lymphocyte, which provides humoral immunity, that means based in the blood, and those are what we normally consider to be antibody or immunoglobulin-mediated immunity against extracellular pathogens, their toxins, and other environmental substances. Now, that by no means are those exclusive definitions. T cells do deal with infectious agents all the time. This includes bacteria and viruses. And B cells are often triggered, or they are induced or activated by T cell responses. So it's only B memory cells that are particularly set up, and there's also surveilling cells, the IgA series, which are set up to be able to deal with pathogens that are just show up because of the infection court. But we'll talk about comparison and contrast with that, don't worry. Now, there's a third group of cells involved in the presentation of antigen to T cells. This is really critical in understanding acquired immunity. These are called APCs, which is antigen-presenting cells. They include the cell types dendritic cells, macrophages, monocytes, and the B cells themselves. Okay? So there is a subclass phenotype functional diversity when it comes to the cell lineages in the immune response, even within the lymphocyte armamentarium. Now, APCs, and we talked about this already in this arc, take up predominantly protein antigens, carry out partial proteolysis, and then bind the peptides to the major histocompatibility complex molecules, which are on the surface of those cells. And they display those presented antigens on that surface where they can be recognized and bound by the antigen receptors on T lymphocytes. T lymphocytes are identified by a surface cluster of differentiation. That's what the CD stands for. And those are called CD molecules. One of them is CD3, and it's comprised of two major groups, CD4 and CD8. So you're going to hear about people calling T cells double positive or double negative. That has to do with the CD4 or CD8 cluster of differentiation molecules on their surface. And of course, that's going to be a descriptor of a functionality that is going to diversify from CD4 to CD8, as we shall see. Now, T lymphocytes are identified by the CD molecules I just said. The CD4 cells display helper activity, so they're called T helper cells. And there are also a population of cells in turn are subdivided in at least, probably 4 or 5 different lineages, all from CD4. TH1, TH2, TH17, and TH9, and then the Tregs. The Tregs suppress the activity of that other cluster of other 4 T helper cells, right? Of course, we talked a lot about this. And each of those 5 TH cells, each of those T helper cells, T lymphocytes, produce their own nested set of cytokines. The cytokines are secreted proteins, glycoproteins, but they're not antibodies, not immunoglobulins, but they do engage and fortify and communicate an infection and then indeed an inflammatory response if they are pro-inflammatory cytokines, or they suppress the inflammatory response if they are anti-inflammatory cytokines. Now they're also functioning with another class of proteins called chemokines, which are also secreted from T lymphocytes and other cell types too, of course. And those tend to form a chemical gradient for cells to track where a site of infection or inflammation is. Okay. So it's very general. I'm just giving you very general descriptors, and I'm sure you've heard this before. Now, CD8 lineage, CD8 T cytotoxic population are known as, is a second major group of T lymphocytes that function killing target cells directly. They are comprised of TC1 and TC2 subpopulations, and they have similar cytokines to the TH1 and TH2. So they also produce cytokines. But these cells directly kill, for example, infected cells. Okay. Whereas the CD4 population, on differentiation, they work to communicate how to kill an infected cell. But they don't kill the infected cell directly. Now, there are a lot of different crossovers between all these T lymphocytes now. And we have even innate like T lymphocytes, which I talked about at great length last month, and I really recommend you go back and listen to that. So there's a repertoire grows all the time, but this is just a very, very simplified version of the 2 different major classes of T lymphocytes. Now, here's a good cartoon of this. This comes from Biochemical Pharmacology, published now 9 years ago, almost exactly. So you've got progenitor cells, okay? And they can differentiate into what's called a DN1, and that N stands for -1. So they do not carry CD25 on the surface cluster differentiation, but they do carry CD44, right? Then they can become both positive, okay? That is, they're still negative in the sense that they do not possess CD4 or CD8 because they're on their way to synthesizing that. So it can still be called double negative because they're not double positive until they express those clusters of differentiations 4 and 8. That's just the way the nomenclature goes, so you have to accept it. Now this is old, but still used in the literature, so you have to understand this if you're going to understand how immune cells function. So progenitor cells in the bone marrow colonize the thymus. They commit to natural killer cells, that's up here, it's a different lineage, right? NKs, or T cell lineages. And they undergo progressive differentiation from double negative to double positive to single positive CD4 or CD8, which just gave you a generalized description of. The immature double negative thymocytes can be further divided to DN1 to DN4, as you see here. They're differentiated, okay? Where either both, here's where 44 goes away and 25 is expressed on the surface. Here's where both of them are gone, and here's where you start to make a transformation of what's on the surface of these cells, the CD4 and CD8, and you also get the T cell receptor called the beta. Now there's a T cell receptor alpha type, T cell receptor beta type, and you can talk about whether or not they are low or high in density on the plasma membrane on the surface of these cells. Okay. So gives you a rough idea what's going on here. Progression from the DP stage to the S single positive stage can occur through an immature CD8 single positive ISP. Okay, that's what single positive is going to be there. An immature CD8 positive single positive cell intermediate. Okay, get the idea? Now there's a couple of other lineages here. Now there's the natural killer cells that come right after the progenitor cell. Those are going to go out and they're going to act a little bit like an innate immune cell. They're going to go out looking for cells that look damaged, presenting something on their surface like an epithelial or endothelial cell. It doesn't have to be a cell of the immune response, of the innate immune response. And they go out and they'll kill it directly. Right? NK cells are very, very powerful, and they take care of business without any necessary cell surface membrane communication. Then there's the gamma delta T cells. That's a different class of T lymphocytes. I talked about those at great length, again, about 2 months ago. We'll get back and we'll talk to them later. To say anything that's not a lot is saying nothing, so I'm just going to call them gamma delta Ts. They're a specialized class. They're really important in infections. Don't get me wrong. NKs and gamma delta Ts are. They're just not in the bona fide major class lineages, which we're looking at this afternoon. Right? The other thing that this diagram points out that I want to point out too is that you have positive and negative selection. That means that these T cells, as they're maturing in the thymus, they're being presented with autoantigen. And if they react or are activated by autoantigen, they are destroyed, right? So they carry out an apoptotic pathway so that those cells never make it out into circulation. That's because T cells are very powerful. If you have T cells that recognize self-antigen, it's going to give you an autoimmune disease, which could kill you at the farthest extreme of that status. So you want to eliminate, negatively select means to eliminate. You also want to positively select those cells which can make it through this transforming lineage, determining that they're going to be double negative or double positive, because those are going to eventually differentiate ultimately to all those different lineages. For example, in the T helper population, 1, 2, 17, 9, and the T regs. So they have to have been recruited, they have to have been educated, and they have to work correctly before they're allowed to survive. That's why you have that selection process. Very important and critical whether or not these T cells function correctly. There's a paper published a couple years after that in Hematologica, and you can see the citation there. What does this paper tell us? I'm giving you background. So what I do in authentic biochemistry before I start giving you some serious new data, right? And then I bring things together as the architectonic. Okay, so this is published in January 2013. You can see the citation. Many disease therapies require allograft event deliveries, okay? So this is when you need a transplantation, for example. So transplantation of an organ or of new progenitor cell. Let's say you are suffering from lymphoma, right? You might not an autograft, you may have an allograft. It comes from another source, not yours, right? Sometimes also genetically engineered differentiated cells like CAR T cells, which we've talked about also in the past, and we're going to bring up again soon here, soon meaning in a couple of weeks, when we're discussing the manipulation of aging cells. So those are just three common reasons why disease therapies require allograft, right? You need organ replacement, you need progenitor cells because you have some hematopoietic disease, for example, and also genetically engineered differentiated cells, which can do a whole host of positive effects on organs and other tissues. Okay. Those introductions, however, often involve a sharp rejection-based anti-transfer immunogenic response. It's well known, right? It's called rejection, right? Organ rejection. You think about getting a lung replacement or a liver replacement. So the acute responsive graft versus host disease remains a frequent complication of allogenic hematopoietic cell transplantation, as used in lymphoma and leukemia patients. So I told you the blood-borne cancers primarily is what this paper has been talking about. So protopathogenic donor T cells are dependent on molecular attachment of small GTPases. This is the cells themselves, internally synthesized from, of course, cytoplasmic ribosomal machinery. Small GTPases to the cell membrane, mediated by farnesyl or granulation of those proteins. And we've talked about this just last time. Don't worry, I'm going to show you those structures again eventually, but those are prenolipids. Farnesyl is a C15 prenolipid, and geranyl is 210, so that's a 20 carbon prenolipid. Covalently attached to proteins such as these GTPases that function to control the activation or the deactivation of T cells of various populations. So now you've got a covalent modification leading to the triggering response in T cell immunity. Right? That's what we're getting at here. Okay. So because of that, you might understand that this GTPase, because it's necessary for T cells to be activated, right, as agents of an immune response, that it could be that if you could fine-tune that placement of that GTPase in the surface membrane of those cells, which is going to be a major signal transducer, right, so that cell becomes activated, you might be able to modify or suppress that rejection phenomenon if you can suppress T cell activation rather than killing the T cells, which there are other drugs designed to do that. Right? Okay, you get the idea. So covalent modification then becomes a target. So a farnesyltransferase inhibitor and a GG transferase inhibitor on acute graft versus host disease, right, plus an anti-cytomegalovirus T cell response and graft versus leukemia activity, we were all investigated in this paper in the mouse model. Treatment of the mice. It's basically just giving you the gist of the paper. Treatment of the mice undergoing the allogenic hematopoietic cell transformation, with transplanting being the ultimate goal, with either farnesyltransferase or geranyl inhibitor, reduce the histological severity of graft versus host disease, right? And significantly prolong survival. So you're trying to remedy lymphoma and leukemia, right? So that's the host disease. And you're trying to decrease the severity of the graft disease. So it's a matter of ratio, right? So you've got a disease you're treating, that's the therapy, and you're using these FTIs and GTIs, right? GGTIs to block the rejection of the allograft. So you want to do a ratio of severity of the graft versus the disease itself, right? And you have to try to tip that balance. Obviously, you want to decrease the severity of the graft response, but greatly enhances the activity of whatever's going on with T cells. That's a fine line, right? So you get the idea. Now, mechanistically, farnesyltransferase inhibitors and the GGTIs resulted in a reduced allo-antigen-driven expansion of the CD4 T lymphocyte lineage. Okay. In vivo treatment led to increased thymic cellularity and polyclonality of the T cell receptor, which means it basically doesn't function very well. Okay, of that receptor repertoire, because remember that's dealt with and generated by recombination mechanisms, right? Just like the B cell immunoglobulin pathway. By reducing thymic graft versus host disease. That's how it functions, apparently. In this paper, that's what they were able to demonstrate. Okay? These effects were absent when squalene production was blocked. Now, why do they tell you that? Well, if you know your cholesterologenesis pathway, squalene metabolism is way down the road, down the trough from the synthesis of C15 and C20, farnesyl and geranyl pyrophosphate, which are intermediates in cholesterologenesis. So if you block squalene, you might say, well, maybe this event has nothing to do with those C15, C20 precursors in that pathway. And they demonstrated that's not the case. Because if you block squalene synthesis, it's not affecting the farnesyl and the geranyl pathways. You see? They're still abundant and activated, which means that they hit their target right on. They were over the target and they were able to impact it, right? That's what it means. Now, let's go with the future discussion. Sorry for all this writing, but I'm summarizing the entire paper. And I want to give it a lot of credit. I'm not going into the data of all these papers because they're papers to build the foundation of where we're going, you see. If I went through all the data of all these papers, each discussion would be 45 minutes long, and it would take me, because I do 10, 20 papers, maybe within one video lecture, it would take me 10 or 20 hours to get through it. And even I might get a little bit concerned I'm getting a little bit away from the main topic. But probably not, but still in all, I can imagine it's what it would be like for some people. All right, so let's go back to this. The farnesyltransferase inhibitor and GT transferase inhibitor did not compromise, look at this, CD8 function against leukemia cells or reconstitution of T cells that were subsequently responsible for anti-murine cytomegalovirus responses. So this is really good. It's affecting the CD4 lineage, which is what they think is being targeted by blocking the farnesyltransferase and the geranyl transferase activity. They don't think that CD8 differentiation to the ultimate targets that they're going to be working on are going to be affected, at least not differentially and not potently differentially, by the elimination of the farnesyltransferase activity or the geranyl transferase activity. It's going to be modifiers of GTPase proteins, which modify the activation quotient of the CD4 lineage, not the CD8 lineage. Okay? So, and plus, it didn't have any effect on getting the cytomegalovirus because that's a CD8 response. Right? So the T cell lymphocytes still can kill the infectious agent cytomegalovirus through all the major pathways we're able to still have at their disposal, but at the same time, they're able to eliminate that allograft rejection pathway. Right? So this is all good. This is all really good. In other words, the specificity and the finesse of the targeting is pretty well set up. So in summary, they observed an immunomodulatory effect, that's what you would call CD4 versus CD8 is immunomodulatory, of inhibitors of the farnesyltransferase and GT transferase on graft versus host disease with enhanced functional immune constitution. That means we were able to push actually more CD8 activity, which was able to eliminate the disease itself, because remember, lymphomas and leukemias have to do with it's a cancer, a proliferation, right, of leukocytes and lymphocytes. And so that's still functioning, even though you're inhibiting. So inhibiting that means you're destroying the cancer, but you're not preventing the mouse anyways from getting its reservoir of T lymphocytes necessary to ward off a cytomegalovirus, for example, which would be an infectious agent. So this is all very good, right? And that means you're able to tip the scales, what we're saying here. All right. So the inhibitor that they used here was called tipifarnib. It's still being used, as you'll see, and that's the one that is the farnesyltransferase inhibitor. Using that in patients and the potent reduction of the graft versus host disease in mice, which is again all this paper is doing is in the murine model. Farnesyltransferase and GGT inhibitors could help to reduce graft versus host disease significantly without having a negative impact on immune reconstitution once it all comes back to play. Now, here's a paper that was published much more recently, as you see. Science Reporter published this year, just a few months ago. You can see the citation. So what are we talking about here? Well, this is now several years later, 7 years later, and we're still talking about using farnesyltransferase, transferase inhibitors. Now, why is that significant? Because science is always trying to reach a new goal. It's trying to find, because that's why science exists in not so much in the theoretical field, but in the combinatorial medical field, right? Which is trained, the medical field is trained by physiology and biochemistry and genetics, right? So if you want to be in the biomedical field, you want to know the papers that are being published on the foundational understanding of cellular activation, again, in the very myopic version of discussing T lymphocyte activation. You want to be sure that years later, after it's been described and introduced in the murine model, people are still using these inhibitors and still looking at them to fight two major diseases, right? What are they? Leukemia and lymphoma, at the same time trying to suppress that rejection phenomenon. So that's why I brought this paper forward. Now, what does this paper tell us? T-cell lymphoproliferative disorders, leukemias and lymphomas, are heterogeneous neoplasm characterized by clonal expansion of mature T lymphocytes. Clonal means CD4 versus CD8, and then all those sublineages we talked about. And that has to do also with TCR, the T-cell receptor recombination, right? In bone marrow, blood, or elsewhere, right? Like, for example, in lymph glands. They include peripheral T-cell lymphomas, PTCL, and T-cell acute lymphoblastic leukemia, or T-ALL. Right? Those are the two major forms of these lymphoproliferative disorders in human. Right? Now, PTCL, that's a lymphoma, accounts for about 10% of all non-Hodgkin's lymphoma, which is a different type of lymphoma, and comprises a set of disorders that includes peripheral T-cell lymphomas not otherwise specified. That's what the NOS stands for. Peripheral T-cell lymphoma not otherwise specified. Angioimmunoblastic T-cell lymphoma, or AITL, is another subgroup of disease. Anaplastic lymphoma kinase positive, or ALK kinase positive, and ALK negative forms of anaplastic large cell lymphoma, also known as ALCL. And finally, enteropathy-associated TCL2. Okay, so these are all major lymphomas. Now, despite the subtypes being very different at the molecular level, their treatment is basically similar, consisting primarily of CHOP, which is cyclophosphamide doxorubicin. That's the common chemotherapy, right? That's what CHOP is. Cyclophosphamide doxorubicin. Right? That's what CHOP stands for. Vincristine and prednisone. Those are the most common ways to treat these diseases, right? Now, there's also a drug called etoposide, right? And all of those follow a similar regimen. I'm not going to go into all of the pharmacology of those drugs because I've done it in the past, and we can do it later if someone sends me an email and wants me to spend 5 minutes on it, I'd be glad to. So therapeutic responses to these treatments show a current general 5-year survival rate of only 10-30% of the diagnosed patients. That's not a very good survival rate. And a rapid disease severity progression in some of those subtypes of some of those diseases. So those drugs, the CHOP drugs we just mentioned here, are just not doing what we need. We need to be able to get a much higher disease-free survival rate, specifically disease progression and survival rate. That's what we're looking for with these diseases. We don't want to have disease progression. We want to see that disappear. We want to see survival rate increase. Right? So T-cell acute lymphoblastic leukemia, the TL, is one of the major lymphomas, okay, or leukemias, excuse me, is an aggressive hematological malignancy characterized by the accumulation of immature T lymphocytes, known as lymphoblasts, in the bone marrow, right, before education. And they're also found secreted in the peripheral blood. And in contrast to the PTCL, right, clinical progress has been achieved with a 5-year survival rate that's much better. So with the leukemia, the T-cell acute lymphoblastic leukemia, a very close friend of mine died of that years ago when we were both in our 20s. He was 27 when he died from that. There was no known cure for this. This was way back, this would have been 1984 when he died of it in Utah. But now we have a 5-year survival rate because we do have drugs for this. We talked about the drugs for TL. And I don't want to go into them right now, but it has to do with translocation phenomena between two chromosomes. At any rate, we do have a reasonably good survival rate for 5 years out. We just don't for those lymphomas. That's what we want to get to. That's what this paper is trying to tell you. Classification of T-cell lymphoma, lymphomas, leukemias, it's complex, requires the integration of information about clinical, pathophysiological, and genetic factors, and epigenetic factors, I would say. Morphology of the cell types, immunohistocytochemistry, flow cytometry is used, cytogenetics, of course, as markers, and the biochemistry with relatively poor prognosis of PTCL4. So the peripheral T-cell lymphomas, as opposed to leukemias, are the ones that are still, we haven't gotten very far in a long time. Right? So that's why people are looking at potential targets, new targets. I just told you 7 years ago, what were we looking at? The farnesyltransferase inhibitor, and the geranyl-geranyl-transferase inhibitor. Exactly. Now, deregulated survival pathways and mutated genes have been partially characterized in the PTCL with mutations affecting the T-cell receptor signaling, appearing as one of the more common pathogenic mechanisms in several of the TCL entities. TCL, remember, those are lymphomas. Mutations in genes related to TCR co-stimulation and signaling were recently reported in a range of TCL entities, such as cutaneous T-cell lymphomas, those are the CTCLs, the PTCL, NOS, which we just talked about, not otherwise specified, the ATTL, and the ATLL, which finally is the adult T-cell lymphoma leukemia. All right. Now, after that brief description, let's discuss prenylation as a factor in T-cell proliferative disease. Aberrant activation of oncogenic RAS signal transduction. RAS genes are often implicated in tumors. Aberrant activation of the oncogenic RAS signal transduction is a very frequent finding in the PTCL and the TAL, okay? Both in the lymphoma and the leukemia, with mutations in the RAS family genes inducing a constitutive activation of RAS mitogen activated protein kinase, the MAP kinase pathway, which of course activates several downstream effectors that play a role in regulating a variety of cellular functions, including growth, survival, and ultimately differentiation, terminal differentiation. The FTIs were designed to disrupt RAS farnesylation because that protein is gifted with that farnesyl group to lock it into the membrane, interdigitate it, I should say, into the membrane. And the memory localization is therefore necessary, it's been shown necessary for RAS function. That's why we want to inhibit the protein's farnesylation that'll block its localization to the plasma membrane. Now, some phase 1 trials using that same compound, tipifarnib, right, it's the inhibitor of farnesylation, have demonstrated anti-neoplastic effects in solid tumors and in leukemias, with phase 2 trials promising early survival when administered as a maintenance therapy in patients in remission. Remember, this is brand new stuff. This is 2020. Okay? Present study, the paper we're going to be looking at, evaluates the effect of inhibiting the farnesyltransferase with tipifarnib in a panel of 25 TCL and T-all cell lines. This is a T-cell study, okay? Enabling them to determine the therapeutic value of the farnesyltransferase inhibitor in those lines and to identify biomarkers that could predict the response to the drug and to measure the dynamic effects of the viability, the apoptosis of the cells that are treated with the drug, cell cycle, if it does progress, and then gene expression, what genes are turned on or off. Those observations could facilitate the development of personalized therapy for individual patients, either suffering from lymphoma or leukemia. The selected panel of 25 cell lines that they looked at included cell lines derived from a wide array of T-cell lymphoproliferative disorders from humans, including the ones that we were just talking about. T-all, CTCL, ALCL, and ATLL. So, 25 cell lines. This is a cell line paper. Right? Now, they're going to key on 3 of the most sensitive cell lines there. The RPMI 8402, the SUDHL1s, and the junkets, okay? Or jerkats, excuse me, the jerkats. Now, you can look at the color of these bars. So how does this graph work? It's an IC50 on the y-axis, cell lines on the x-axis. Very common, basically an inhibitor curve assay on cells, right? Some of the cells are from the T-all, some are from the ALCL, and some are from these 2 other lymphomas, right? So 3 of the most sensitive, and so the lower the bar, the lower the IC50, that's how much drug you need to knock out half those cells. Sorry, let's go back. Very low amounts, nanomolar amounts. So you want to use cells that are very sensitive to the drug, and so they chose 3 of them. The RPMI, which is a T-all, right? That's a leukemia. The SUDHL, which is an ALCL, that's a lymphoma. And the JERCAT, which I think is also another leukemia. Looks like it is. Okay? So you get the idea. While they're using those cell lines, plus they probably were really good in culture. I'm guessing they were easy to culture and easy to show that the drug at very low concentrations had a really strong effect on them, knocked them out. So 3 of the most sensitive cells were selected to test cell. And what do they test after using the drug? Viability, that is after you remove the drug and see whether or not the cells can be put on fresh media and then divide. That's viability. Induction of apoptosis, do the cells then respond to the drug by killing themselves via programmed cell death? And then by flow cytometry, just using surface markers to determine whether or not, the total number of cells. These cell lines differ in terms of subtype mutational level, so there are some mutations in these lines, of course. The JERCAT and the RPMI 8402 are derived from T-all, so that's the leukemia, while the SUDHL1 is derived from the lymphoma. Okay. So we know this because I just told you. Now, you can look at cell viability, and I would say, except for this one line here, this SUDHL, now let's go back. All right. Okay. All right. Now I know it. All right. Here we go. Arrows go up and down. Isn't that amazing? So SUDHL, remember, that's the unique one, right? So these other 2 are T-alls. The other 2 are leukemias. So this SUDHL is the lymphoma. That's a lymphoma. I want to bring that back and explain that to you. So, cell viability, right? Is it affected by, okay, so this is cell viability. Is this affected by the drug either at the IC50, that really low nanomolar amount, or doubling the IC50, which is how these kinds of pharmacotherapeutic analyses are done. Right? So doubling the IC50, you should get doubling the effect. Cell viability doesn't look like it's really affected that badly, right? So there's the IPM, RPMI, and there's the JERCAT. Those are the two leukemias. Yeah, okay. It's better than a DMSO, right? And there is some significance. You can see some significance between the control and at least at the IC50 level of treatment, maybe, right? And there might be some difference here even between the control, that is just a carrier DMSO, dimethyl sulfoxide, and then having double the drug, right? But the one that really showed, I think, the best response was a lymphoma, just on viability. Okay, so cell viability, induction of apoptosis, and tipifarnib sensitive cell lines are what we're looking at, and these percentages are all going to be relative to the DMSO, right? So now that's what you've got down here. You've got induction of apoptosis. Now let's take a look at this. Down here, you want the histogram to get bigger if you're getting a good effect. And it looks like, indeed, compared to the DMSO, a whopping increase in the amount of this annexin B being expressed. That's a sign for apoptosis, right? Induction of apoptosis. And in all three cell lines, in the RPMI, the JERCAT, and also in the lymphoma, you've got, as you add the farnesyltransferase inhibitor, you get a great increase in the induction of apoptosis. DNA synthesis, not so dramatic, but similar, right? You get a decrease in DNA synthesis, that is replication of the chromatin, as you increase the amount of drug here, but it's not statistically difference between the IC50 or the double the IC50. And pretty much it's similar here, although the JERCAT does tend to show a slightly better response when you double the drug. But certainly, once again, lymphoma seems to be the most sensitive, which is kind of interesting. Interesting from the point of view of the pathophysiology of the disease, right? Lymphomas versus leukemias. So the percentage of total DNA is exponentially growing. Tipifarnib-sensitive cells, the probabilities of those isolated. They used the Fisher's t-test, the exact t-test, just a statistical design, the way they did this. So you can see that the drug is working. It's causing programmed cell death. It's diminishing cell viability, and at least with a couple of these cell lines, with the dose response that we're looking at, you were getting a reduction of DNA synthesis, which of course is a sign of whether or not it can replicate. If it can't replicate its genome, those cells aren't going to be viable. Next generation. These are just tests. This is what you do in cells in culture. So, tipifarnib does not change the T cell phenotype. This is critical. What does that mean? They study the possible changes in Th1, which produces interleukin-2, Th2, which produces a pro-inflammatory cytokine interleukin-4, Th17, pro-inflammatory cytokine IL-17, and the Tregs, the transcription factor there is FOXP3, something I spent a lot of time talking about in the last several months. Those phenotypes, by analyzing and changing the expression of their markers in 8 cell lines, you can see that they're represented there between leukemias and lymphomas, including the 3 that we just looked at. Cytokines that allow the characterization of inflammatory, non-inflammatory, and regulatory subpopulations were all measured, and what they determined whether there were no phenotypic changes when the cells were cultured with tipifarnib at the IC50 or even at double the kill, 50% kill concentration, right, inhibitory concentration. That's good. So it's not altering T cell phenotype, because if you use a drug and it looks like it's enhancing apoptosis, it looks like it's decreasing cell viability, it looks like it's decreasing the ability for those lymphoma or leukemia cells from replicating because the DNA replication levels are decreased. That's all good, but if it also then modifies the lineages of all those T cells, like what kind of cytokines are producing, what kind of transcription factors are expressing that thing, that's really going to make it a mess, right? Because now you're corrupting the normal T lymphocyte immune response. You don't want to do that. And it doesn't look like inhibiting farnesylation did that, at least in this cell-based research. Now here's a real take-home message here. These pathways are all down-regulated by tipifarnib, and the bars are all shown at the log level. of inhibiting these pathways. There's the jerkit, there's the RPM1, and then the N. The reason that the blue is at the end of this kind of graph is because they have the highest level of downregulation. It's a log scale here, right? So what kinds of pathways are being turned down and therefore inducing what we just saw, the desirable response? Well, some of them have to do with ribosome activity, ribosome machinery, splicing of messenger RNA, oxidative phosphorylation was negatively impacted, right? And then you can go down here, DNA replication, we know that, it was inhibited. Here are nucleotide biosynthetic pathways. I love this, purines and pyrimidines. Ubiquitin-related proteolysis is also diminished. Those are all down-regulated by this farnesyltransferase inhibition. You can go through here. It's chronic myeloid leukemia, whether or not a direct effect on that disease. Amino acyl transfer RNA biosynthesis, you need that for translation, obviously. Protein export is impacted. These are all turned down. And then in some of the regular standard metabolic pathways. Pyruvate metabolism, for example, lactic acid synthesis, acetyl-CoA synthesis, oxaloacetic acid synthesis. Fatty acid metabolism is also downregulated. DNA sensing pathways, this has to do with toll-like receptors. Pentose phosphate pathway is turned down. Homologous recombination is turned down. It's one way. Homologous recombination is a repair mechanism, recall. Also important for gametogenesis and for syngamy, but mTOR signaling also seems to be negatively impacted. These are the main pathways affected by the inhibition of the farnesyltransferase. So many things we could start looking at, and obviously you're going to have to, because down the road, using these inhibitors can indeed affect whatever else the immune system is doing in these individuals. Right? Of course it can. Okay. Now, T-cell leukemias and lymphomas account for between 10 and 50% of all the lymphoid tumors, and in most cases are characterized by an unfavorable prognosis, with only about a quarter of the patients remaining alive 5 years out. Okay. Given the important role of T-cell receptor signaling in neoplastic T cells, they decided, the researchers, to study the RAS-MAP kinase pathway, hence the farnesyltransferase inhibition. The members of that family to elucidate the genomic basis of lymphomas and leukemias with aberrant activation of the oncogenic RAS signaling transduction, and that's what they're trying to impact with the farnesyltransferase inhibition. Tipifarnib could be potential therapeutic option in T-cell leukemias and lymphomas, given the majority of the cell lines were sensitive to it, 60% of them were, at clinically achievable concentration. So the IC50 there, remember, was below 100 nanomolar. It's really good. And that's within a 96-hour exposure to the drug. There is an association between ERK, that kinase, which is a major component of these proliferative pathways, lymphoproliferative pathways, measuring phospho-ERK, right? That means it's being activated, and that means that lymphoproliferation is proceeding. And so when you have those active, you have an increase in the farnesyltransferase inhibitor sensitivity. Conversely, REL-B, which has to do with cell cycle, probably reflects activation of the alternative NF-kappa B pathway. That in itself is a pro-inflammatory system. That was associated with tipifarnib resistance. That's curious, right? And it suggests that mutational status of a protein called Notch1, which is a transcription factor, and the activation of the ERK kinase could serve as potential biomarkers to look at the relative rational activity and that means sensitivity of the tipifarnib, and perhaps the positivity of REL-B by immunohistochemistry could be a biomarker of resistance to the drug. I was just looking at that REL-B using flow cytometry, for example, or in situ immunohistochemistry. Since there appeared to be a link between ERK activation and tipifarnib sensitivity, this group focused on the RAS-MAP kinase pathways, we said. Other T-cell leukemias, lymphomas related pathway, T-cell receptor, JAK-STAT pathway, NFAT pathway, and NF-kappa B, which are all linked when they are dysregulated in those immunoproliferative lymphoproliferative diseases, leukemia and lymphoma. 7 sensitive and 3 resistant cell lines harbor mutations in the RAS genes. No surprise there. 8 sensitive and 3 resistant cell lines harbor mutations in the RAS guanine nucleotide exchange factors, the GAFs. That's all part of that GTPase complex. But the differences were not significant. 5 sensitive, 38%, and 2 resistant, 25% cell lines harbor mutations in directly the RAS GTPase activating protein GAP genes. That was also a non-significant effect, although it did show trend. The mutational state then of RAS-GAP, exchange factor, RAS-GAP, right? That's the activating protein. Those genes, mutational state of those genes was not associated with the farnesyltransferase sensitivity. Okay? Mutational state of all of the RAS-GAP and ERK activation of REL-B negativity, and you can see that they're very low p-values, right? So it looks like the effect of this farnesyltransferase on the potency of inducing an inhibition of immunolymphocytic proliferation did not have to do with mutations in several of those key oncogenes. It appeared, though, the TCR pathway, the G protein coupled receptor pathway, the GPCR, and the checkpoint, I told you some of this was checkpoint stuff, G2M phase, right, when you go into mitosis, that's at the end, they were correlated with greater sensitivity to the farnesyltransferase drug, while NF-kappa B activation, either by TNF-alpha, interferon gamma, these are all pro-inflammatory cytokines, and the interferon alpha response, that works against infectious agents like virus, ribosome and p53 pathways were correlated with resistance to the drug. Now that's interesting. That means those pathways are less susceptible. That means directly to farnesylation. Of course, that's what it means. They performed an RNA-seq study in 3 of the most sensitive cell lines, ones we just talked about, and more than 300 differentially expressed genes, including 24 that change their expression consistently in all cell lines. And we were just looking at those pathways, right? So it's becoming clear that selective alterations, their take-home message here, of key prenylated downstream signaling enzymes, such as the GAFs and the GAPs, and membrane communication networks, which is the result of their activity, may be useful in targeting of these hemolymphocytic diseases. So right now in 2020, inhibiting the farnesyltransferase is important. So you can see that all these years later, it's still considered an important system to inhibit. And the more we do this kind of work, the more we learn about it. Now, I'm going to leave you with this last slide for this afternoon. And we'll start talking about this paper. This is a critical review of biochemistry and molecular biology published 2 years ago. Now, I have been detailing how post-translational protein prenylation and prenyltransferase inhibitors, we just saw that paper, you saw the data. The data looked good, looked like good evidence. Those may be, the FTIs may be therapeutic agents for oncogenesis and for metastasis. Right? I've also reported the scientific literature is well represented for demonstrating how protein prenylation has continued to grow. In addition to cardiovascular disease and cancer, prenylation can be associated with aging, potentially senescence-associated cardiovascular and neurological diseases, along with autoimmune disorders, diabetes, metabolic syndrome, infection, and inflammation. So all of those pathways you saw that were negatively impacted feed into all of these pathophysiological stages. Besides glioblastoma, which I told you we're eventually going to be getting to here, and general neurodegeneration, AD or Alzheimer's disease is also linked to protein prenylation. There's a whole prenylome associated with the neurodegenerative disease, AD. Now that'll be a topic that holds discussion from this paper will be a topic for an Authentic Biochemistry podcast, probably a video, but maybe an audio. And that will be coming up in the weeks ahead. I'm going to get into that, okay? Haven't done it yet. We're going to get into it. So that takes us back to where we were, talking about the fact that we're using carbon in the form of pyruvate. You utilize pyruvate to make acetyl-CoA. Acetyl-CoA forming citrate. The TCA cycle allows for the citrate to be translocated out into the cytosol because there's an adequate amount of NADH and FADH2 being made. This leads to production of citrate cytosolically, which will specifically enhance the amount of the MVA pathway. There it is up here, methylmalonic acid pathway, making one of the major intermediates, the farnesyl pyrophosphate, which is used as a farnesylating agent. You're also going to make other isoprenoids, such as geranyl pyrophosphate and cholesterol itself. Right? Some fatty acids are also going to be made from the ATP citrate lyase. Of course, the OAA can feed back into the system after that reaction. That's the ATP citrate lyase reaction there. Remember also you're using amino acids to drive this in T cells. Remember I told you that? So glutaminolysis generating glutamate, then finally alpha-ketoglutarate, a reductive carboxylation to citrate, and that citrate feeding right into isoprenoid biosynthesis and fatty acid biosynthesis. And that's where we were last time. So I'm going to end this now because the next category of discussion is going to be in the steroid response element binding proteins, which are going to lead to our understanding of how these genes are controlled at the level of expression. What genes? Farnesyltransferase, geranyl transferase, as well as those MVA core enzymes on the way to make those intermediates, as well as things like ubiquinol and, of course, cholesterol, and then all the downstream processing of that very important, significant membrane lipid. Okay, so we're going to stop here, and we're going to move on to this lecture. This next lecture will probably come tomorrow or maybe Tuesday. I'm not sure if I'm going to do it as a video or an audio, but you'll be receiving this. So we're going to continue on this discussion of T cells utilizing the MVA pathway to control their activation. I just showed you some clinical information that suggests that if you inhibit this specific farnesylation and maybe the geranyl of certain proteins involved in membrane-associated signaling, such as with the ERK kinase and the GTPase activity, you may be able to decrease these lymphoproliferative diseases. At the same time, you may be able to can still have an active T lymphocyte immune response because of the differentiation of all the T lymphocytes. All right. So with all of that in mind, I'm going to leave you now and hope you have a pleasant Sunday afternoon and say bye for now.